So without further ado, we're going to start with Aviv Regev.
Slides don't show. Awesome. Thanks a lot for the opportunity to speak here today. I have a disclaimer that I'm not a human geneticist. I'll do my best. But I also have disclosures. I'm a co-founder and equity holder in Celsius Therapeutics, and I'm an SAB member in Thermo Fisher, Neogene, and Cirrus. And I'm going to talk today about cells, which are the basic unit of life, and that come in many different types and shapes and form, which we've historically classified in biology based on different features, from their structures to their molecules. Cells are also a key intermediate between genotype and phenotype. I think we saw that extensively already today. And in particular, although all cells in the body carry the same genetic variants that confer disease risk, the diseases that would actually occur depend on the cell types that express those genes or that are impacted by their gene product. And so knowing our cells is actually essential to understand how genes that cause disease act. And of course, the only problem with that is that we don't actually really know all of our cells and what are their molecular characteristics. And there's an additional problem, which is cells that have already been categorized in many different ways based on their structures or where they're located or what functions they have. And so it would be lovely if we had some joint map or unified set of coordinates. And I think it's well accepted that gene expression levels and other types of molecular profiles can provide these unified coordinates such that each cell will be a point in this high-dimensional space. But as Eric already alluded to earlier today, until recently, we couldn't really fully pursue this kind of idea because we only had these measurements of expression profiles in bulk, also known as the fruit smoothie. But in the last few years, because of breakthroughs in single-cell genomics, it's now possible to really measure expression, chromatin, and other kinds of profiles in very large numbers of individual cells. And there's also been a lot of progress in spatial genomics approaches that are beginning to allow us to do this in tissue. And so today, I'm going to mostly focus on single-cell genomics that has one of these curves that we love in genomics, where things grow exponentially. And as a result of looking at cells in these high-dimensional spaces, over the last 6 or 7 years, it's become quite clear that we can learn a remarkable amount of biology from these profiles. We can learn the cell types and states, differentiation and other temporal processes, gene programs, and even the physical locations and interactions between cells. And I think we all have this assumption that this could give us a better vocabulary by which to describe and understand disease and answer questions like, where do disease risk genes act? What types of cells are disrupted? Which cell programs might be changed? What are the functions and modules of the cells? How are the communications between cells affected? And ultimately, what is also the effect of drug? And so after they cut me 3 minutes, I'm going to skip some slides throughout the talk, but I will try to cover four different aspects. The first, I'm going to describe to you very briefly the human cell atlas. And then I'm going to talk about how we can use the approaches of a human cell atlas in order to try and build atlases of complex common diseases, looking at the example of ulcerative colitis. Then I'll turn into the questions of how single-cell genomics can also help with deciphering gene function at scale. And I'm going to try and close with a few opportunities for ICDA. So first, the human cell atlas. The mission of the human cell atlas is to create a comprehensive reference map of the types and properties of all human cells as a basis for understanding, diagnosing, monitoring, and treating health and disease. We kicked off at an international initiative, just like this one, in fall 2016, after some planning that happened earlier. And it's since progressed to an atlas building endeavor that has biological networks that collect and analyze data and has a very elaborate data coordination platform for housing the data of the atlas. It's a rapidly growing international community. It's open to all and that are willing to adhere to its mission and values. And we take enormous pride in the fact that we have 1,500 members in HCA from 64 countries and over 1,000 institutions. This is a big effort across a very diverse set of people. And so it's guided by what we call a roadmap, which keeps evolving, that spans the different systems, tissues, methods that we use as scientists, but also the ways in which we coordinate things, the ethics and the standards that are needed to assemble the atlas. Together, we aim to map the whole human body. In our first version of the atlas, our focus is on 14 organs and systems, and each is represented by its own biological network of scientists, and they include things like the nervous system, the gut, the lungs, the liver, the reproductive system, the immune system, and so on. And just to give you a glimpse of the data collection efforts, this is a slide from a few months ago, and so just as in these 6 example tissues, we've already collected millions of cells from hundreds of individuals around the world. In the process, we gained both access to, say, very rare cell types we didn't know exist, but also confirmed the high reproducibility of the findings. And so for today's purpose, though, my goal is not to talk about HCA, but to really show how the power of HCA-like approaches can be applied in the context of common disease. And I'm going to focus on the example of ulcerative colitis, because ulcerative colitis is a form of inflammatory bowel disease, and IBD is really a poster child of human genetics. And this is where GWAS found, hundreds of new risk variants that pointed us to new processes like autophagy and new pathways, and can ultimately lead to new therapeutics. But I think it's fair to say that in most cases, we don't really know the cell of action of the variant, and we don't know its function, and we don't know its mechanism. And so we were hoping that having an atlas would help with that. And in trying to address this, we partnered with Ramnik Xavier, who's right here and from NGH in the Broad, to collect what is now a single-cell atlas of 365,000 cells from 12 healthy donors and from 18 individuals with ulcerative colitis. And we collected both inflamed and uninflamed tissue from the patients and had replicate sampling and ended up with 68 samples. And so we can start looking at first what we call the training set, the initial data set, which is 120,000 cells. And we see that the cells partition into 51 different subsets. Within the epithelial cells, we can recover the differentiation of those cells. Within the immune cells, we find cells in the myeloid B and T cell compartments, and we also find many different kinds of stromal cells. In fact, we found every cell type that we thought should exist, and many others that we didn't know existed, except for neutrophils and for enteric neurons that are both rare, and we know have the issue surviving dissociations, and I'll come back to them in a minute. But for now, we can just look at this cellular composition. The first thing that you see is that it's not static. If we compare across healthy, uninflamed, and inflamed tissue, we see that the composition is actually dramatically remodeled. And so it's dramatically remodeled to the extent that the ulcerative colitis colon can have cells we didn't even expect exist. So I'll just give you 3 examples. Within the epithelial cells, we find these cells that we call M-like cells. They're not supposed to exist in the colon. They're supposed to exist in the small intestine, but we see them in ulcerative colitis. In the stroma, we find a new set of fibroblasts that are associated with inflamed tissue, so we call them inflammatory fibroblasts. And amongst the T cells, we see an expansion of CD8 T cells that express IL-17, which is supposed to be expressed by Th17 cells. And so now that we had this really rich catalog of cell subsets, we wanted to ask the question that was already posed here today: can we see where the UC genes from GWAS actually are expressed? And so partnering with Mark Daly and with Ramnik, we looked at the genes that they've already mapped. We see them here in the columns, the 51 subsets in the rows, and we find that most of the UC risk genes are only expressed in specific cell subsets in the mucosa. And this was partly mirrored, actually, by doing FIWAS on the UK Biobank that, again, implicated sets of these GWAS genes in traits that were related to specific cell types. Now I take you back to this expression map, which is very satisfying, but you might also notice that there are some genes on the right-hand side that do not appear to be substantially expressed in any subset. And so this may be for many different reasons, including the fact that we're missing some cells. I told you that we didn't have the enteric neurons, but also that we only looked at the mucosa of the tissue, and that's just one layer of the colon. And so to address both of these problems, we turned to another method we developed called single nucleus RNA-seq, and we used frozen resection materials from another 10 healthy individuals to build an atlas of deeper layers that recovered many other cell types, including adipocytes, as well as interstitial cells of Cajal, which are super rare, and enteric neurons. And when we add those additional cell types, we can relate additional UC GWAS genes to the cells, including to specific subsets of enteric neurons, but also, for example, 5 genes relate to those adipocytes. And this shows how important it is to have comprehensive catalogs rather than partial ones. Now, given this expanded tissue depth, we can also ask about cell type specific expression of risk genes for early life or late onset CNS disorders that we know have a concomitant gut dysmotility. And this means that we will be profiling, and because we actually find their specific expression in this map, it might mean, to a question that was asked earlier today, that you can profile a more accessible tissue like the ENS instead of the inaccessible CNS. But going back to ulcerative colitis, we can now look which cells are highlighted by the analysis. And we find that some subsets are enriched for GWAS genes. For example, remember those M-like cells that are not supposed to be in your colon? These rare epithelial cells actually are not just expanded in ulcerative colitis, but they have a preponderance of GWAS genes expressed in them, and those GWAS genes, their expression is further induced in ulcerative colitis. And so now we wanted to see if we can also use these data to predict the function of GWAS genes, for which there is not a good annotation. For example, a gene with an inspiring name like C1orf106, which is a GWAS gene where at the time the function was not known. So one way of predicting gene function is to ask which other genes co-express with it. And in the past, we would do this by comparing one cell type against another cell type. And if we do it, we would get something like gut epithelial genes, because C1orf106 is expressed by epithelial cells. But we now can look only within enterocytes, which are the cells that express C1orf106, and we find that it actually co-varies with tight junction genes. So we can postulate that it has a function in tight junctions, and conveniently for us, Ramnik's group recently shown that it regulates the stability of epithelial adherence junctions. So now I wanted to generalize this lovely approach across all GWAS genes. We map the genes to cell types. For each GWAS gene, we look for genes that are co-varying with the cell type, and then we're going to iterate this with different GWAS seeds until we converge on a set of modules that connect GWAS genes to other genes. And what happens when we do this is that most modules are enriched from multiple GWAS genes at a time, so that in fact 10 such modules together span more than half of the UC GWAS genes in a cell type specific manner. And so now we said, could we use this behavior, now that we know it exists, to nominate risk genes in a multi-gene locus? That is, if one characteristic of the GWAS genes is that they aggregate together in modules, can we revert the reasoning and prefer candidates that co-vary with other GWAS genes or with other candidates in other regions? And you have to iterate through this. And so we wanted to test this by taking cases where the causal gene is already known in a multi-gene region, thanks to Mark and Ramnik's work, hide its identity, and see if we can use either expression or covariation with genes in other associated regions to predict the causal gene. And it actually gives us a twofold increase in accuracy compared to a null where we just would pick a gene at random. What's interesting is that it's true only for genes that are in UC or UC and CD, but not for genes that are CD alone, that emphasizes that you need an atlas from the disease context to do this. And now we can, of course, take the regions that are not known and make new predictions. So next, we asked whether we could use this modular structure of the programs and see if we can help with additional types of genetic questions. For example, what about interactions? We can't reach significance by testing every pairwise interaction because that's like 24 billion tests, but we could maybe introduce a prior, such as the structure of these GWAS genes that fall into modules. So we decided to take a look at this problem by combining our full atlas with an IBD exome cohort of 22,000 individuals. And to do this, we rebuilt modules in two different ways. We did cell type specific modules only of GWAS genes using variation across all 365,000 cells. And we did program modules that are based on covariation of the GWAS genes within a cell type using the GWAS genes as a seed. And then we started to look for ways of using this for subtler signals. Now, this is very much work in progress, and I told you I'm not a human geneticist, but we still think that the results are intriguing. So, for example, we can look for implicated cells by performing a burden test on each of the gene modules between controls and disease samples, and look for a high number of high consequence coding mutations in the module. And that this flagged some potentially significant differences in modules that's related to some subsets of CD8 cells and enterocyte progenitors. Then we can also look at genetic interactions, and we can explore this in two different ways. In one way, we take genes within the same module, and we ask, do they interact with each other? And for example, we can find an interaction between LAL-RB1 and NOD2, both in dendritic cells. It's more difficult if you want to look at interactions across the different modules. So we first identify modules that as a whole might interact with each other by their aggregate signal, and then we look at pairs of genes between them. And one example would be an interaction between epithelial and stromal cells, and specifically between OR5L2 and PKD1. I'm not going to elaborate on the polygenic risk score, and so I'll just summarize this part by telling you that I showed you a glimpse of the cell atlas of ulcerative colitis, which is a complex disease atlas. We built it from involved and uninvolved tissue in patients, as well as comparing to the healthy reference from HCA. A relatively small number of individuals provides a really robust catalog, and we found that most of the GWAS genes are expressed in a specific cell subset, and as we expanded the cells, we got better information, so you need comprehensive atlases. The GWAS genes also form these co-varying modules with each other and with other genes, and those account for a large proportion of those genes in a small number of modules. They helped us predict GWAS gene function, improve our prediction of causal genes in a multi-gene region, and then we can incorporate this structure to try and identify subtler genetic signals. And there's much more work that could be done with these types of structured information in the context of the polygenic risk score, patient stratification, and single-cell QTLs. And so for the next part, and much more briefly, I want to tell you a little bit about how we can relate genes to cells and programs, because single-cell genomics can actually help a great deal in this as well. And for this, we rely on the single-cell profile as a very high-content readout for screens. This is based on a method that we developed several years ago called Perturb-seq that combines CRISPR screens, pooled CRISPR screens, with single-cell RNA-seq as the readout. And so very briefly in Perturb-seq, we start with a pooled library of vectors that carry guide RNAs and barcodes, but the barcode is going to be expressed as an RNA itself. Then we grow our cells. I have 2 minutes left. Then we grow our cells, we sequence them by single-cell RNA-seq. They get a cellular barcode that's attached both to the guide and the gene and allows us to really identify what's going on. And so we have done this in the past. This is a screen in primary immune cells called dendritic cells with LPS, and we see beautiful structure both in the affected genes across the perturbations and in the factors, in this case transcription factors that we modulated. We can do it for combinations by just increasing the infection, and so we get multiple perturbations per cell, and we can infer genetic interactions in this way. And really the question is whether screens like these can work to assess the functional variants. And I'm going to try and show you extremely quickly a couple of examples. And so the first one is to consider loss of function variants that affect complex phenotypes. In this case, de novo protein truncating variants implicated in ASD, which we expect would impact brain development. To make a long story short, we do it by infecting embryos in mice. We inject them with a pool of lentivirus at E12.5, wait until 7 days after they're born, and then assess the impact of each variant. Because we have single-cell RNA-seq, we don't actually need to know what phenotype we're looking for, and there's at least 2 that we can look at. We can ask if the composition of cell type is affected. Turns out only 1 affects the composition of cell types. But then we can also ask whether there are gene programs that are affected, and we find that 15 of the 35 genes that we tested significantly affect the 6 programs across 4 cell types. In the second example, we can do it also with coding variants that are not loss of function, which I'm illustrating with cancer. In this case, we assayed by Perturb-seq a pool of barcoded variants in KRAS and P53, along with common missense variants from EXACT. I'm not going to show you how we did it, because time is shorter than planned, but I will tell you that it works really well to classify wild-type, gain of function, and new variants into existing categories for both KRAS and for P53. I'm going to skip this. I want to make only my closing points. So first of all, that Perturb-seq is a family of methods that couples perturbations with high-content readouts. The original application was with CRISPR loss of function. We can apply it in vivo. We can apply it for coding variants. We can apply it with other types of readouts, like ATAC-seq or with optical readouts of cells, and it can actually scale. I had a slide for that, for millions of cells, and to start going after combinations. In terms of the key opportunities, I think there are 3 I want to highlight. The first is the building of a reference atlas for disease tissue. Everything from building mini cohorts to single-cell QTL studies, building a new observational vocabulary of programs, and then predictive models. The second is these massive high-content pooled screens for function. Again, developing the techniques, deploying them across systems, using them to develop another vocabulary of programs that is based on our own directed perturbations and hoping to develop predictive model. And the last is to develop a new analytical framework that really takes these functional modular insights and brings them together with human genetics. And with this, I'll conclude. These are just acknowledgements from people in my lab that went throughout the slides. Thank you very much.
Okay, thank you very much, Aviv. We're moving on to the next speaker.
Absolutely, next speaker.
Shendure from University of Washington. Can you please help me with that?
I thought my talk was packed, but it will be less packed. So that was great. Okay, so, oh, sorry. Okay, so let's see here. Okay, great, thanks. Glad to be here. We have 12 minutes, I'll try to stay on time. Just as kind of a backdrop to this, just some high-level points. In this V2F2M space, right? So there have been, we're sitting on top of tens of thousands of clear associations. It's worth taking note that there have been relatively few clear success stories still in this mapping of variance to function to mechanism, but there have been some that are solid. But each one, by and large, has been a major undertaking. So on one hand, I think it's important to be sober about how challenging this will be, kind of looking forward into a major effort like this. But at the same time, of course, it's important to move forward. And I think that related to Eric's point, I think earlier this morning about infrastructure type projects, what can we do? What are the kinds of things we can do to try and make this overall process easier, right? To sort of grease the wheels, so to speak, right? So systematic resources for facilitating these kinds of goals that could be broadly useful. So I'm gonna talk about, I'm just gonna briefly touch on 3 different projects from our lab that relate to this, right? And obviously are kind of smaller than the kinds of things that you could imagine, but kind of give a sense of maybe what's possible if this is the kind of thing that a few graduate students or postdocs can do. So in the first base, gold standard catalogs for variant effects. This is work from Greg Finley and Leah Sarita. So we and many others have over the years been pursuing these kinds of massively parallel functional assays of testing large numbers of variants in parallel in cellular or in vivo assays and using sequencing as a readout for the relative effects of variants in that assay. Increasingly, one can do this with CRISPR, right? So the kinds of perturb-seq assays that Aviv mentioned, also actually introducing individual nucleotide variants in parallel into a system of interest, right? And so here, for example, what we've done recently is to use what we call saturation genome editing to introduce many possible variants of BRCA1 exons in parallel into this HAP1 cell line, which is dependent on BRCA1 function for survival, right? And so I won't go through all the details of how we did this, but the goal, of course, was to try and see if we could use these sorts of functional measurements to adjudicate variants of uncertain significance, which more on the rare side have been a challenge in the clinic. And so focusing on 13 exons, which is by and large where clinically relevant variants, missense variants for BRCA1 fall. And I won't go through all the details of how we do this, but basically it's a survival assay where we introduce many variants at once into a given exon. We see which cells die and which cells live, and from that we can compute scores. And then we can look at whether these scores actually correlate with what we know clinically to be the case for a small number of these variants. And importantly, this is saturation, right? So it's near saturation. We're hitting about 97% of all possible SNVs. And I think, for me, kind of one of the things that I really like about this kind of approach relative to the classic approach for variant functional analysis, and this has a lot of analogies with the world of genetics where, one-off association tests for your candidate gene of interest were replaced by more systematic scans where you get a view of the entire distribution of effects. So similarly, even though we may be only interested in a handful of variants in this distribution, we gain a lot by being able to visualize the entire distribution of effects and to see where things fall relative to other things, right? So in this case, with these particular exons, the distribution turns out to be bimodal, with nonsense variants falling on one side, synonymous on the other side, and missense splitting in between. And then if we look at variants that have been, quote, adjudicated by bodies like ClinVar, we have very good agreement with this assay and what's used in the clinic. So, but only a handful of these variants have actually been observed in the clinic, so this becomes the kind of data you can use for VUS resolution. Okay, and so that's again, this is in rare variant genetics, but I think there is a lot of power here that could be applied, including to regulatory variants. So here's just an example. This was not with genome editing, but rather with an MPRA assay, but we were able to measure over a series of experiments the regulatory effects of around 30,000 single nucleotide variants in 20 disease-associated variants. This is one, a sort1 enhancer, which is associated with, which has been implicated by GWAS, and we can look again at where the individual implicated variants fall relative to other variants, so on and so forth. Okay, so second type of approach here, so that could be a sort of an infrastructure type map. So this is work from Molly Gasparini. So projects like ENCODE, I think, are a great model in the sense that they've generated these terrific resources that are broadly used by our community and many other communities. One challenge has been, we have many candidates based on biochemical nominations, but a lack of equivalent functional kind of analysis. So we've taken inspiration from GWAS or even QTL studies in the sense that, you can think about what an association study is. You're effectively reusing a population to perform many association tests, right? The people constitute a scrambling of genetic variation. So analogously, rather than people, you could imagine having cells. And so, of course, cells are going to be genetically homogeneous if you're doing this like with a clonal cell line. But you can replace naturally occurring genetic variation, and instead of that, effectively rely on multiplex CRISPR-based perturbations, right? So here, we're using high multiplicity of infection, lentiviral infections of cells to basically have a unique assortment of perturbations in each cell. And so then we can perform the same kinds of association tests that one does in QTL studies, using many of the same statistics and logic, and effectively looking at different combinations of cells that bear any given guide targeting a particular enhancer. So we're testing many candidate enhancers against many transcripts, much like in a QTL study, you test many possible naturally occurring variants against gene expression changes across the entire genome. So in sort of the proof of concept work here, we evaluated the effects. We functionally perturbed 5,000 enhancers in the K562 cell line, sequencing around 200,000 cells for their transcriptomes, and then performed association tests. Again, we're analyzing this like QTL studies. It's a QQ plot, right, where you can see a gross inflation of significant p-values when we are targeting enhancers as opposed to targeting or as opposed to non-targeting controls. And just drilling into one of these in detail. So here on the three this is the NMU locus, and down on the left is the gene itself. And we're evaluating a series of candidate enhancers in the vicinity. And we have a cluster of sites about 50 kilobases upstream where perturbing any putative enhancer in that block results in gross downregulation of the gene, but perturbing other elements that fall in between does not, right? So from this kind of approach, so one of the things that I was particularly excited about was, we talk a lot about enhancers and where they fall relative to the genes that they regulate, and we often cite examples of, sort of the extreme examples. The enhancer was a megabase away, right? So this is an approach that you can get a less biased view on the endogenous distribution of enhancer TSS distances in the genome, right? And so when we look at it this way, we're basically testing hypotheses across a 500 kilobase region, but the vast majority of associations are between enhancers and promoters that are about 24 kilobases apart. So it's a median distance, right? So picking the nearest gene doesn't always work, but it often works, right? And this is kind of an outside check of that assumption, which is often used. Okay, one more example, and this relates to some of the things that Aviv talked about. How can we leverage single cell atlases to further knowledge of the etiology of common disease? So we, a couple years ago, generated an atlas not of gene expression, but of chromatin accessibility, and not in humans, but in the mouse. So this is about 13 or 14 tissues from an adult mouse where we've measured chromatin accessibility collectively in around 100,000 cells. We're able to define about 85 cell types on the basis of these data. So, the kinds of things you can look at with this include, that are different than single-cell RNA-seq data, include differential accessibility. So we built a catalog of around 400,000 elements that are differentially accessible. So here looking at various subtypes of astrocytes, and you can see that only one of them has this particular element accessible. Here looking at various subsets of endothelial cells, plausibly corresponding to different organs. And we can see some specific accessibility there as well. So this represents an opportunity, one could imagine, to try and intersect with GWAS signal and see, as I think Eric's talk also alluded to, if we can find the particular cell types or subcell types that are of principal relevance to various common diseases. So the challenge here, of course, is that we did this with mouse for our chromatin accessibility data, but we only have human GWASs. But it turns out you can just lift over the SNPs based on synteny, and it still works surprisingly well. So here we're just taking human SNPs, lifting them over to the mouse genome, and then computing overlaps with differentially accessible peaks in our atlas. And so what do we see? So the rows here are 85 human cell types and the various chromatin accessibility profiles, and we're intersecting that with the columns, which are various GWAS studies. And so just to give a few examples, Bipolar disorder, you can see that we get enrichment for a variety of excitatory neurons. That's where the maximal, quote, overlap is with bipolar. But we look at Alzheimer's disease, not a single neuron comes to the forefront, but rather microglia, right, which is now relatively well established. So taking advantage of the summary statistics that Ben Neal kindly put on the web from UK Biobank, this is now the extension of that approach to all of around 2,500 traits. And this is all a browsable matrix, and there's just some fun anecdotes. So things are largely what you expect, but there are some interesting things in there. So just as an example, gout primarily associates with cells of the kidney proximal tubule, emphysema with venous lung endothelium, and systolic blood pressure with heart arterial endothelium. And so kind of we can potentially nail down not only the organs, but also the particular types of vasculature that are of relevance. And then a fun one, the pain when you walk is in your mind, not in your legs, and that kind of thing. So I'll stop there, and just some closing points as we're changing people over time.
Thanks, Jay. So Jimmie Ye, next one.
Great. I'd like to thank the organizers for giving us the opportunity to share some of our work. It's good in the first session to see some of the questions that came up about looking at gene by environment or gene by gene effects. In fact, my lab has been somewhat obsessed about looking for context-specific effects for quite some time, and specifically the type of contexts that we're interested in are cellular state. And so here's an example of this. Many of us have done these bulk expression QTL analyses where we compare the expression level in bulk between individuals of different genotypes, and you'll find some differences in expression. Of course, we know, as Aviv demonstrated earlier, that in bulk, it's sort of a heterogeneous population of cells. And if you look at cell context, in this case of two different cell types, you observe that the genetic effect is only really observed in these triangle cells. And so a consequence of that is if you look at the bulk expression, the effect size is much smaller due to this phenomenon called Simpson's paradox, because there's a possibility that you will have variation in composition between individuals. And so we want to look for effects like this, and hopefully it doesn't take too much to convince all of you that this is going to be important for understanding to ultimately reveal disease mechanisms. But understanding cellular context could also be really useful as a proxy for environment, which could potentially be modified in common disease. But this task is in general difficult in bulk and potentially unperturbed cells. So my favorite example of a gene by environment interaction, in this case a gene by infection effect, is in the gene ERAP2. And ERAP2 has been associated with a number of autoimmune diseases, and most recently it's also associated with anti-PD-L1 response in bladder cancer. Now, ERAP2 goes back about 5 million years. There's still only 2 haplotypes in the human genome. There's 40 SNPs in perfect LD and about 100 SNPs in almost complete LD. And the conundrum is that the major haplotype, or about 56% of the chromosomes in the world, encodes for a version of the gene that's degraded by nonsense-mediated decay because of a splice variant. And so the result of this is that ERAP2 is one of the strongest cis-UQTLs we'll ever detect. Individuals who are homozygous major basically don't produce any transcript. Now, why would something like this be under balancing selection in humans with such a long stretch of LD? And what we did when I was a postdoc in Aviv's lab is we infected monocyte-derived dendritic cells with virus. And in response to virus, something remarkable happens. You basically rescue the expression of the gene. Each dot here, by the way, is an individual. And you'll see that the effect size changes from about 2 log scale to about 0.61. Still statistically significant, but this is one of the strongest interaction QTLs that I've ever seen. And so we're spending a lot of time in my lab to try to figure out exactly what is happening with this haplotype in response to virus. Now, how do we look for G/E effects more broadly? And so one of the things that we did when I was at the Broad is to sort of take a somewhat biased approach, because we only could do so much at the time given the technology, which is we took peripheral blood mononuclear cells, white blood cells, and profiled 2 different cell types, and also taking innate immune cells and adaptive immune cells and activated them with a number of cell signaling molecules. And so I'm not going to bore you with the results in detail, just to say that we detected genetic and ethnic specific effects, and many of them are cell type and stimulation specific. But the lessons learned from this is that bulk sequencing is expensive, laborious, biased, and we cannot estimate cellular composition. And so my lab has since turned to massively parallel single-cell RNA sequencing. And the way most of these methods work, whether or not you're using droplets, wells, or Jay's method with Sky, basically you're indexing a cell using a physical or virtual compartment, which is illustrated by this circular barcode. And then after sequencing, we get this giant matrix, and then we reduce that matrix to 2 dimensions for visualization purposes, sort of similar to principal component analysis of genetic data. And so you can imagine that if we got this data on hundreds of thousands of people, we can potentially map genetic effects across a number of cellular contexts very efficiently. But this is really expensive to do. And so when I started my lab, we wanted to figure out a way to reduce the cost for single-cell sequencing at the population scale. And the insight here is that there's enough genetic material contained in the transcriptome of a single cell for us to uniquely identify that cell. And so what we do now in the lab is we just pool cells from tens of people together and flow these cells into an instrument like 10x Genomics at much higher collision rates. So for us, we're basically sequencing cells at about 5x the concentration that's recommended. And then we wrote a piece of code called Demuxlet that, taking prior genotyping data, we can identify what droplets contain multiple cells in it. So we remove doublets, and that reduces the cost per cell. But we're also able to demultiplex, which is assign each cell correctly to the donor of origin. And so that reduces the cost per sample. In return, we get this much larger matrix. On average, our sort of per run, we're getting about 3 to 4 times more data than a standard 10x run. And so MuxSeq and Demuxlet, what we demonstrated in our paper is that demultiplexing is very accurate. We make 1 mistake out of about 4,000 cells. That reduces per sample cost. The ability to detect doublets allows us to increase the throughput of 10x Genomics and other types of single-cell assays, and that reduces the per cell cost. For us, sequencing 1,000 cells is about $200. For 5,000 cells, it's about $600. And I should say that about 90% of that cost now goes to Illumina. And so as sequencing cost drops, our cost for sequencing single cells will also drop. Importantly, for the type of things that geneticists are interested in, we minimize batch effects, and we can also start with frozen cells and nuclei, which will sort of minimize experimental manipulation. Others have demonstrated that genetic multiplexing can also work with a single nucleus RNA sequencing, site-seq, where you simultaneously measure protein and RNA, as well as single-cell attack-seq, has a star on it because we're still figuring out sort of some of the parameters around that. And we also have a version of Demuxlet called FreeMuxlet now that works without prior genotyping. So for the rest of the talk, I want to show an example of the application of MuxSeq to profiling a human disease. This is systemic lupus erythematosus, which is a systemic autoimmune disease that's about 66% heritable and disproportionately affects women. There's no new approved drug in the past 50 years. And peripheral blood mononuclear cells, or circulating immune cells, are a particularly good model for SLE, since many SLE-associated loci implicate a number of innate and adaptive immune genes. And there's this known type 1 interferon signature expressed in PBMCs of lupus patients. So here's how we did the experiment. We had 2 arms, a cross-sectional and longitudinal arm. The cross-sectional arm, we sequenced about 750,000 cells. On the longitudinal arm, we sequenced about 250,000 cells. So it's about 1 million cells total. And the way we did it is we multiplexed 16 donors at a time. And for subsequent runs, we always had 2 healthy controls that were replicated that allow us to estimate the performance of our demultiplexing algorithm. And just as sort of a point of reference, if we actually wanted to do this for bulk RNA sequencing, let's say that there's roughly 9 cell types in circulating blood, and we got about 200 donors, it would be 1,800 samples. And it's actually not cost effective compared to multiplexing single-cell RNA sequencing. And so we're not going to do that. So this is what a projection of a 1,000,000 cell looks like. Again, these are two arbitrary dimensions. Cells that are phenotypically close to each other are closer in distance in this projection. And we can call out about 10 different cell types. These are very broad lineage cell types. And we can go within each of these and call out specific activation states as well. And so what we can do with our data is we can demultiplex and in fact generate a patient-specific immune profile. And the ones on the right are the controls, the ones on the left are SLE patients. If we just look at the distribution of immune cells, I think qualitatively you can see a difference that in a particular population of monocytes seems to be enriched in lupus patients, as well as some CD8 T cells. So I'm going to take a step back for a second, and I'm going to take the two monocyte populations and group them together and take that proportion and multiply it by the number of white blood cells per volume of blood. And I'm going to take the 4 or 5 lymphocyte populations and do the same thing. The reason I'm going to do that is we can now calculate an abundance of monocytes and lymphocytes from blood. And this is a value that's recorded in the complete blood count for all of the patients since they're UCSF patients. And so we can get that information from the EHR. And so we really wanted to assess how quantitative is our data. And really, to our surprise, the correlation of monocyte and lymphocyte counts were really good between single-cell estimates and what's reported in EHR for the same donors. For lymphocytes, it's about 0.97. For monocytes, it's about 0.88. So if you think about what we did, we took frozen cells, thaw them, threw the cells back together, sequenced to demultiplex, call cell types, and then got this sort of aggregate estimate of monocyte and lymphocyte abundance. So we were pretty surprised and encouraged by this. This means that we can do some genetics. Of course, we also asked if there are differences between patients and controls, and we reproduced this known phenomenon called lymphopenia. So lupus patients have a reduced abundance of lymphocytes. Of course, with single-cell data, we can also go in and ask what are the precise cell types that are depleted, and it's the CD4 positive T cells that are depleted in lupus patients. But there's expansion of CD8 T cells and slight expansion of B cells. So we took each of those 8 different cell types, or 7 different cell types, and did an EQTL analysis. And along the diagonal here, I'm just writing down the number of cis-EQTLs that we detected in each cell type. And above that, the upper triangle is a cross-heritability analysis where we're able to show that cell types that are related in lineage share also their genetic architecture. If we now ask what are the cell type-specific regulatory elements, they're also enriched. That's encouraging. And if we look at the SLE GWAS loci, they're enriched for CD4 and CD8 cis-EQTLs. And on the right-hand side, they're just some examples of cell type-specific EQTLs. So I want to end with this example. And so I talked about context initially as this discrete change if you're comparing stimulated cells versus unstimulated cells. But in vivo, context could be a continuum or degree of activation. And so we're going to sort of have a change of axis on this plot and show it this way on the right, where we're showing stimulation on the x-axis and gene expression on the y-axis, and then genotype is differently colored. And then in some cases, we can actually measure the degree of activation quantitatively. It turns out in lupus, I mentioned earlier, there's this interferon signature that's been known for about 50 years. And that's a quantitative measure of the degree of interferon in the blood. And so we can estimate that interferon and ask, is there a gene-by-environment interaction? That is, can somebody's genotype determine the correlation with the expression of the gene with the interferon activation? And really, to our surprise, we were able to find such an example in ApoBac3B, which is an RNA editing enzyme that's supposed to have antiviral effects. So individuals who are heterozygous, the correlation between ApoBac3B expression and interferon is negative, and for individuals who are homozygous, it's positive. So what's next? I'll just close here. We are embarking on this next project where we're going to try to create a reference of what healthy immune profiles look like. The goal is to sequence 10 million cells, 400 samples cross-sectionally across 4 ethnicities, and 600 samples longitudinally. We're going to measure RNA and protein, and in some samples, chromatin. These patients or healthy donors are going to be broadly consented, so we'll have available genotyping data. And we're developing a cloud compute and sharing platform, and this is a CZI-funded seed network across UCSF, Columbia, the Broad, and Johns Hopkins. We're also part of this EQTL-GEM meta-analysis consortium, and that's going to be across 4 continents. And my first approximation there is that they're going to be about 2,000 samples. I'm not going to talk about the applications. We can discuss some of this maybe over the discussion session. Thank you.
Thank you very much, Jimmie. We're moving along. Next is Ramnik Xavier from the Broad Institute.
Okay, I want to thank the organizers again for this opportunity, and what I thought I'll try to do in 12 minutes is to briefly introduce some of the concepts that we've used to try to understand biology behind genetic variants, and then propose a few examples of studies to consider for ICDA. So broadly, we think in simple terms, so we view sort of genetic susceptibility genes as a path to finding mechanisms of disease, and protective factors as finding mechanisms of health. And I'll give you a couple of different examples along this way. So the first observation is autophagy. So prior to the genetic observation, there was no link between autophagy and Crohn's. A coding variant was identified in the gene, making it the culprit gene. What was interesting is that despite it being an autophagy gene, the coding variant had no effect on basal autophagy. It only functioned on selective aspects of autophagy. Over 3 years, we went on to identify many features of this variant working in very different cell types, so protecting against bacteria, affecting the genesis of Paneth cells, promoting healing of wounds, keeping bacteria away, but most importantly, identifying a caspase variant, cleavage site within the coding variant that made the coding variant more susceptible to disease. The clinical implications of this was a study done in collaboration with Skip Virgin and Thad Stappenbach when we looked at patients who had the coding variant and abnormal Paneth cells. Their time to surgery was much shorter than individuals who did not have the coding variant and who did not have Paneth cell abnormalities. So a direct clinical application of this finding. This led to the observation that there are many variants within the 250 plus loci. Could we find additional autophagy-associated genes within those loci, so currently not annotated as autophagy-associated genes? To do this, Cara Larson ran a screen to ask for genes that affected bacterial replication. She identified something like 20 different genes that were within coding variants, and that led to the biology. I'm going to tell you about one story where we identified a gene called GPS65, which was a G-couple receptor. It has a coding variant listed up here. And as you move from a healthy variant to a coding variant to a knockout allele, you can see how the phenotypes of that variant change, all dependent on its ability to acidify lysosomes. So clearly, multiple effects as a result of this variant. More recently, in collaboration, we've gone on to identify screens to identify small molecule correctors of that variant, and I've begun to sort of characterize one of those variants. So that's one example, autophagy, multiple genes, effects on IBD. The second is, what about one variant, many disease? So here's an example of a transporter, SLC39A8, that's associated with risk for Crohn's disease as well as schizophrenia. We've gone on to map structurally where this variant might be located. So the clue to this was this same transporter is found in bacteria, so we could very rapidly map the structural variant. And what we've identified is that this variant affects glycosylation in a manganese-dependent manner. Obviously, the studies that are ongoing in the lab are to understand the effects on gut barrier function, but could it mean that glycosylation is a much broader process in the central nervous system that leads you to defects in clearance of poorly glycosylated proteins and risk of schizophrenia? The third example is probably the best, where you can find a coding variant that's a risk gene, work done by Manny Rivas when he was with Mark Daly, but at the same time identifying a protective variant. And if we can mimic the function of the protective variant, that would probably be the best therapeutic hypothesis. So we've gone through a series of experiments. So there's a risk variant that increases inflammation, a protective variant that attenuates inflammation. We've identified a binding partner, so an ubiquitin ligase that binds fails to bind the protective variant, thereby titrating its effects on inflammation control. And so clearly the advantage here is that if we can mimic the protective variant, one could find a therapy that's already proven by nature to be safe. So we've taken 3 different approaches. Because of my time limit, I've taken out the data, but using these 3 different approaches, we can find binders that mimic the protective variant. The 4th example that was briefly alluded to by Aviv was the idea of a gene where there is no known function. How do you go about it? And this gene is C1off106. So again, in a study that Manny Rivas led with Mark, identified a coding variant in C1off106. We systematically went about trying to understand the mechanism, so did proteomics initially to identify a novel binding partner that interacted with C1off106. We then showed that expression levels of C1off106 affected the function, but more importantly, if you overexpress the gene, you can correct the defect. The phenotype is localized to the junctions between cells, and you can sort of track this over time, showing that the gut tends to be leaky if you express the variant or you delete the gene. What was more striking was that when we looked in an in vivo model, the junction that was most susceptible to this variant was the junction adjacent to the stem cells in the gut. So suggesting that this could have an important aspect in committing to lineage over time. And then finally, if you look at the variant, it fails to correct that defect. So in the third example, I've given you an example of a gene with no known function, but then taken it down to understanding how it might function in pathways. The 3 examples that I've given you have roughly taken us about 3 years. So is there a way of speeding this up? And I would say yes. So generally the approach has been variant, expression maps, function, in vivo models, and back to human cells. But the unique opportunity here is that as we've sort of embarked on optimizing organoid systems, we can now move from variant to a corrector phenotype within about 6 months. Can we do this? So in the lab, we've sort of taken this approach of sort of mapping innate immune defenses. So we've taken 6 different pathways and asked the question by co-culturing organoids with myeloid cells initially, and more recently myeloid cells with T cells. Can we map these variants? We don't have time to go into detail, but the answer is yes. So for example, in the production of reactive oxygen species or phagocytosis, not only that we've identified the function of the variant, but more importantly identified 3 different regulators of the pathway that has come about because of these studies. So you now know how to control the circuit. And then similarly in nitric oxide, we've identified another gene that sort of regulates this process. In the second example, in collaboration with Stuart Schreiber at the Broad, we asked, can we now find disruptors of that pathway? And we've since identified a molecule can disrupt that KEAP1 and RF2 axis, leading to the production of the antioxidant response. And that's illustrated here. So you can clearly see when you use the inhibitor, you shut down that pro-inflammatory cytokines. So this is an example where we went from genetic variant to a pathway and a rapid use of a candidate gene that could mimic that variant. So what should the future hold? And a few suggestions for ICDA. So this was a study that, again, was led by Manny and Mark, where they looked at the distribution of variants across populations and pathways. And what was striking was the observation that the autophagy pathway was enriched in the AJ population, and the IL-23 pathway was enriched in the non-NJ population. The attraction here is that IL-23 is a drug in the market. So now one can go back into clinical trials with the drug and ask the impact of genetics. But what we've begun to embark on is to try to map the broader pathway of IL-23 signaling. So could we find additional genetic variants that could contribute to this pathway and then go down the path of sort of application from a clinical trial to genetics, so going backwards. The second project is the idea of, can we now start to map coding variants across many diseases? So we've sort of taken something like 20 plus gene risk traits and asked, are there coding variants within those genetic association studies? And the idea here is to create a variant cell bank of cells isolated from these individual patients who harbor these variants, and ask sort of questions where the community could participate, so massively parallel approaches to identify assays, pathways, and map these genes in a systematic manner. The final example is, as Aviv alluded to, is can we move from genetics and try to pay attention to the environment and look at its impact on cells. I think it's important to focus on disease, tap into toxicity, and what might be the outcome. And if you take many of the diseases that were discussed earlier this morning, so all of them have genetic variants, there's clearly environmental contributions to many of these genes, and I would argue that we need disease-focused cohorts to try to better illustrate or dissect the pathways between genes and environment. And this could be obviously extended in a much larger setting and try to collect information on environmental factors, but collect them in the context of disease. I think I'll stop there.
Thank you very much, Ramnik. And the last speaker of the session, before we go to the discussion, is Tuuli Lappalainen from the New York Genome Center.
Much to the organizers for putting together this wonderful meeting and for giving me the opportunity to present. So today I decided to give you a little taste of some of the recent work that the GTEx consortium has been doing, putting together an atlas of genetic regulatory effects across multiple human tissues. So this work is like a few days away from being in bioRxiv together with the main paper and a number of different companions. And I'll give you a little taste of the results, covering maybe like 10% of everything that those papers will cover. Obviously, the general context of why we are talking about these things is building the understanding of biological mechanisms that drive complex disease associations. And here, our focus is really using genetic associations to gene expression and other transcriptome traits to understand GWAS loci, to understand the regulatory code of the genome. Also, there are interesting transcriptome applications to interpretation of the personal genome, which I might talk a little bit if I have time for an extra slide in the end. So the GTEx project set out about 10 years ago to create an atlas of genetic regulatory effects across multiple human tissues. And now with the GTEx V8 release, we're at the point where we have over 17,000 post-mortem tissue samples from about 50 tissues of over 800 donors, RNA-seq data from all the tissue samples, whole genome sequencing from the individuals, and then multiple data types, of course, created from this raw data, looking at transcriptome variation, computationally estimated cell type composition, cis-QTLs, trans-QTLs for expression and splicing, and then integrating these data with harmonized GWAS summary stats for a number of different diverse complex traits. So what do we find when we do mapping of regulatory variants across these samples? So when we look at first just classical cis expression and splicing QTLs, we find tons of them. So we get now to the point where the tissues with the highest sample size is about 600 samples, have over 10,000 genes, well over 10,000 with eQTLs and more than 4,000 with splicing QTLs, really highlighting how extremely widespread this type of variation is and how complete this catalog is at this point. We find also not just a single eQTL or a splicing QTL per gene, but actually for the larger tissues, most of the genes have more than one independent, LD-independent eQTL, and the same is true for splicing QTLs as well, again illustrating this extreme allelic heterogeneity of regulatory effects. When it comes to mapping of trans eQTLs and trans splicing QTLs, the GTEx data is still a little bit limited in terms of power, but there is a large number of genes that actually give us really interesting biological insights when integrated with the cis eQTL data, with us finding cis eQTLs and their kind of downstream trans effects, giving insights into what these genes actually do to regulatory networks. And altogether, now with GTEx V8, we actually have a nearly complete catalog of cis regulatory variants for expression and splicing across human tissues. So, of course, for this audience, the key question is, so how do we interpret GWAS associations with this data? So first, just looking at classical GWAS enrichment in eQTLs, we see both for cis eQTLs and splicing QTLs, clear significant enrichment of GWAS associations in those data. For trans eQTLs, this enrichment is even stronger, as has been suggested before. But what is interesting is that when we analyze eQTLs, not just kind of all significant eQTL and SQTL variants, but hone into the likely causal variants of those loci, those enrichments get very, very strong. 4 in log2 scale, those enrichments are actually pretty similar to some of the coding variant enrichments for GWAS, highlighting the fact that if we are actually able to get to causal regulatory variants, these variants actually have a very, very strong enrichment of GWAS effects. We can, of course, do classical co-localization, predict scan analysis, finding thousands and thousands of GWAS loci with hypotheses of potential target genes, obviously not evidence, but hypotheses. And altogether, these results, together with a lot of the other work that has been done by the GTEx GWAS working group, highlights how these regulatory associations mediate GWAS associations, but also indicating how the challenge of fine mapping needs to be kind of tackled with care. One of the ways how we can actually kind of use that allelic heterogeneity and complexity to our advantage is to look at how regulatory and other types of genetic effects affecting the same gene converge at the GWAS level. So when we look at multiple cis eQTLs, independent cis eQTLs affecting the same gene, we actually see that they tend to have concordant GWAS effects, suggesting that basically, these multiple different perturbations of the same gene contribute to similar traits. We also see a similar trend or similar pattern when we look at independent cis eQTLs and cis splicing QTLs for the same gene, basically asking that if there is a splicing QTL that co-localizes for this gene, is the eQTL more likely to co-localize? And the answer is yes, indicating that different types of molecular regulatory perturbations can have similar GWAS effects. And then finally, looking at genes that have a rare coding variant GWAS association in UK Biobank actually have an enrichment of GWAS co-localization, an eQTL co-localization as well, showing these kinds of joint effects of rare and common variants. And altogether, leveraging on these kinds of complex GWAS signals that come from common regulatory variants, rare coding variants, different types of functional variants, help us to not just only point out the gold standard causal genes for some of these GWAS loci, but also highlight the potential to build allelic series and look at how these different types of variants actually contribute to GWAS. So when GTEx was started, just the idea of building this catalog of eQTLs across multiple different tissues was a hugely challenging goal. But obviously now, at this point in time, we understand that tissues are composed of multiple different cell types, and getting to that complexity and that layer is really, really important. So what we have now done is computational cell type deconvolution, estimating the enrichment of 64 cell types in all the GTEx samples. And we see kind of like patterns that make sense. These methods are not perfect, but they do work. One of the really interesting insights that we got looking at how these different cell type enrichments are shared across tissues and how that compares to sharing of cis eQTL effects is that if you look at this heat map here, it shows the tissue sharing of cis EQTLs across different tissues. Actually, really high degrees of sharing for most tissue pairs, up to like 0.9 correlation. But then when we construct a similar pairwise tissue sharing matrix just based on cell type enrichments, we see that this is actually super highly correlated to the cis EQTL sharing, really showing us that shared effects of regulatory variance between tissues are largely driven by sharing of different cell types, again indicating what a crucial factor cell type composition is when we want to understand and analyze human tissues. The cell type enrichment analysis allows us to map cell type interacting cis-QTLs, both for expression and splicing, looking at basically how the EQTL effect changes as a factor of, or as a, with cell type composition. And we're able to map thousands of cell type interacting EQTLs. These are not necessarily cell type specific EQTLs, because we don't necessarily know if the cell type that we detect is the one where the EQTL effect is active, or whether it's a correlated or an anti-correlated cell type. But regardless, we are able to find a lot of EQTLs where there's clear indication of cell type specificity. And this has interesting implications for a couple of things, but I want to mention a particularly interesting GWAS interpretation aspect. So here we have one locus, just an example of platelet count GWAS locus, where there is, if you go to the whole plot data, there is an EQTL signal in that locus, but it doesn't actually co-localize. There seems to be something else going on. But when we look at an cell type interaction EQTL for neutrophils in whole blood, we actually see a signal that very strongly and clearly co-localizes with the GWAS. And basically what is happening here is that there are multiple EQTLs in the same locus, basically allelic heterogeneity from different cell types. And when you analyze that bulk tissue, all of those multiple EQTLs get smashed together into one signal that you can't really co-localize well. And we see a more general pattern of very high numbers of loci that we are able to co-localize with cell type interacting EQTLs that wouldn't co-localize with bulk tissue. Again, highlighting the importance of being able to have that additional cell type resolution into genetic regulatory variants. So in the spirit of this meeting, I wanted to take just, instead of just listing a summary of what I just said, thinking a little bit of where are we and what happens next. So as I said, we have a nearly complete catalog now of cis expression and splicing QTLs in human tissues in Europeans. But of course, we have tissues, obviously. We need to get cell type. That is pretty clear. We are able to have a very complete map of cis EQTLs, but trans QTL discovery is still quite underpowered, and we have many lines of evidence of the informativeness and importance of trans EQTLs for example, GWAS interpretation. We are looking at expression and splicing and transcriptome phenotypes here, which is not the whole scope of human molecular phenotypes or cellular phenotypes. For example, a TAC-seq is something that has been shown to be super informative for GWAS interpretation. GTEx is mostly European ancestry sample, and now that we are having GWAS done in more and more diverse populations, the molecular interpretation tools actually need to keep up as well. And currently, for example, doing co-localization analysis for, let's say, an African GWAS would be almost impossible because we don't have those EQTL data sets from other populations. And then, of course, mapping just QTLs, that is one approach to do analysis of functional impact. There are many exciting experimental approaches that we can pursue now as well. When it comes to GWAS interpretation, I think that the situation is kind of like a glass half empty or half full. So we have, with EQTL data, been able to create many different resources that have been super useful for GWAS interpretation. But of course, we don't explain all of it. There is some loss of sensitivity, for example, coming from poor resolution in bulk tissues, not having a full data set of all cell types, contexts, ancestries, molecular phenotypes, etc. LD contamination, co-regulation still need to be analyzed very carefully. But one thing that I want to highlight that is important is that the comprehensiveness of the GTEx dataset is really important to avoid naive interpretations of kind of like finding one potential effect and thinking that is the effect. And one thing that I want to highlight after acknowledging many people that have been part of GTEx, in particular François Aguet, who has led the analysis, I want to take just 30 seconds to mention a new approach that we have developed in my lab to basically allow the use of resources like GTEx to interpret personal transcriptomes. So many of you have mentioned getting transcriptome data from various cohort data sets, patients, but we need to be able to interpret those data better. And we have built tools how you can basically take estimates from GTEx to build models of genetic dosage sort of tolerance in different genes, and then put your individuals in that spectrum to identify genes where individuals carry particularly strong effect population outlier genetic regulatory variants. And I'm going to stop there.
Okay, well, thank you very much. I want to thank the speakers for staying on time, actually less than the time originally allocated. So I would like to invite everybody up. We are going to have some time for discussion, and actually, since it went so well and the speakers were so right on time, we're actually going to have a break of 10 minutes, so there's no reason to rush now. So you can breathe a little bit, yes. So please have a seat, and then we're going to have some questions from that you didn't have time to raise before. Yes, right.
Mila Prakonina-Olsen from NCI. So I think there are interesting points here, and one of them is that infection is the most extreme case of gene-environment interactions. So unless you are not just exposed but infected, your genotype doesn't really matter, and this is something to consider. And second, for Jimmie, I think that your case about APABEC SNP, which affects interferon signature, is a very good example of pleiotropy in action, because this variant is extremely known in cancer research because it is associated with genetic regulation of APABEC mutagenesis, right? And I hope you know that it's not just SNP who is doing that, but it's tagging. It's only SNP which is tagging a 30 kb deletion, which completely eliminates the gene. So this effect is extremely important, but no, it depends how you interpret it. And this shows also that it's important to look at these questions from transdisciplinary point of view, because what might be new and surprising in one field is very well known and more explainable in another field. Okay, and one comment for Tuuli. So for GTEx, is it possible really to adjust for covariates when you're looking at these effects? Because sex is important, age is important for any traits. And when you're looking just something which you get from a website, I don't think you have it.
So we can have one individual answering each question?
Yeah, I think it was more a comment. I agree with everything you said. For us, it was more of a comprehensive view and look for individual examples. We haven't followed up on APOBEC3B. I am aware of it's Breast Cancer Association, right?
Well, it's in many cancers, but in general, it's association with APABEC mutagenesis.
Yeah, we would love to follow up to see if the biology replicates.
Let's talk.
Yeah, I'm not sure what the question was regarding age and sex specificity. There is actually a sex sort of specificity companion paper in the works by Barbara Stranger and others. And there are some indications of sex and age interacting EQTLs, but actually a lot of them seem to be driven by differences in cell type composition.
What I mean is that when you're looking at effects on like at least pre-calculated values, it's just straight for the genetic variant, without adjusting for covariates, at least age and sex, right? And you cannot do it unless you download data and do it yourself, right?
Okay, very quick answer.
Okay.
Reply.
Yeah, I guess the question. So some of these interaction analyses will be just like the summary stats will be made available. But obviously, like we can't do everything. So at some point, if people want to kind of like really dig into the data, they will just need to download it.
Okay, thanks.
Question there. Yeah.
Thomas Lehner, NIMH. You all made beautiful case for functional studies in, and it seems single cell is where most of the action is. That's beautiful and very plausible and makes sense to me. But can I ask you to make an argument for other cellular systems, because we have not addressed those, like stem cells, and which are also of particular importance to studying the brain, because we don't have access to the tissue.
To make an argument for
the
what?
use of
For use of cellular models?
For cellular systems in exploring functional biology.
Yeah.
Like
So-
iPS cells?
Yeah.
Yes, for example.
Yeah, so I think both for Jay's talk and in my talk in the second part, admittedly highly rushed, but we got a break from that, so there's something good. You could see that the perturbational assays that are more engineered, meaning any perturbation can be CRISPR, it can be ORFs, it can be anything that you choose to do. You need a cellular system to assess them. And I think currently our cellular models that we apply in those settings are either primary cells or cell lines, typically alone, but in Ramnik's talk you could see that in fact you often would want to do a co-culture or an organoid. And so I think actually cellular models are imperative. One of the values of having high resolution single cell and other types of analysis of human tissues and of cellular models is that you can actually understand their strengths and their limitations. So for example, in an earlier study that Ramnik and us have done in the small intestine, we found that the separation between panet and goblet cells in intestinal organoids is actually incomplete. And you cannot really see it in the bulk assay, but you see it very easily in the single cell assay. If you're interested in panet cells because of the human genetic study, you would better know what the model can and cannot deliver for you. So absolutely, I think they're a very important and integral part of any effort.
Anything else? Okay.
Hi, we know ICDAbio. We are supporting a lot of those large-scale bulk RNA-seq projects. It sounds like the advantage diminish very fast with the single-cell analysis. So the question is there any unique value for bulk RNA-seq we should keep supporting, or we should start to think about switching to a single-cell analysis instead of keep supporting the bulk RNA-seq?
I can take a crack at that. So I think the power for detecting QTLs in single cells, given the same sample size, is a little bit lower than in bulk. We're not entirely sure why that's the case. I suspect some of that has to do with technical limitations in sampling transcripts. So I think, yeah, there's probably still some value in doing bulk analysis or integrated analysis of bulk RNA-seq and single-cell RNA-seq data. Yeah.
I will add 2 additional technical points, I think maybe 2 fine for the forum. The first is that currently most of the single-cell studies that you see are not full-length studies. And so in full-length RNA-seq, you have much more ability to do allele-specific expression, which definitely empowers you and gives you better causality. So that's very important. I think it's a target for the single-cell community to get to the point that they can actually do those things, and that might change the assessment. The second is that we still depend in most of these analyses on an existing pre-assembled transcriptome, and the best place from which to assemble this transcriptome is from deep full-length RNA-seq in the bulk. And I think we should toggle between those 2 things and understand the validity of each of them and think more of hybrid studies rather than of here's the best way of doing you have to use this or you have to use that, but use all of the tools in our arsenal.
Yeah, I would add also splicing to the mixture.
Yeah,
that is not captured well.
That's part of transcriptome.
The GWAS enrichment is pretty similar in splicing QTLs and eQTLs.
Yeah, absolutely.
Yes?
Brian Brooks, National Eye Institute. One brief partisan comment and a quick question. The brief partisan comment is that that ocular tissues are just conspicuously absent from the databases that have been funded, and it would be really great if that were not the case, if decision makers are baking those things in.
Can I interrupt in that?
Yeah.
The first DropSeq paper was the retina, and then it followed by 2 more, and more are coming. And part of the Human Cell Atlas is actually a retina atlas, just to say.
Yeah, yes, so that's fantastic. Thank you very much. That's very good. I'm thinking specifically about target tissues for the common diseases like age-related macular degeneration, for instance, which has large effect sizes, is going to affect 3 billion people as of 2020. And the retinal pigment epithelium is a great system to study. The trabecular meshwork in glaucoma, one of the leading causes of blindness in African Americans, great tissue to study. So that's the partisan comment, and I stand corrected for it.
My point is, for healthy tissue, it's already in the works, and for disease tissue, I think it's this forum's responsibility to emphasize.
And so then the comment is, building on what was just said about using stem cells, specifically iPS cells, are there examples, either from your labs or from the literature, where differentiated cells of any tissue type have, from patient iPS cells, have been leveraged in the way that all of you outlined?
So, I think Steve McCarroll calls it the in vitro mosaicism experiment. I think a lot of people are already thinking about this. We've thought about it in my lab, we just haven't done it. So, for example, it'd be great to pool cells, either reprogram or differentiate them together to eliminate variability across sort of experiments to just explore inter-individual variability. So I think things are ongoing, and certainly on some of our radars, I'm sure.
Yeah, you have Gilad's lab and Jonathan Pritchard, they have been doing these types of analyses as well.
Yeah, and there's also work at the EBI from Oli Stegli and colleagues that has looked at that in iPS cells. It's actually, I think, a posted paper. And there's more. There's work in organoids like that as well, not just in iPS cells.
David Goldman, NIAAA. So a within-cell methodology for eliminating trans effect variability is, of course, differential allele expression, where there's a heterozygous reporter locus, which there often is. So I would ask Tuuli Lappalainen if she sees a value in DAE expression, or do you feel that the bulk sequencing experiments are sufficient? Is that bulk data sufficient, or would we have more refined and accurate cis-EQTLs?
So, I mean, I think I'll just echo what others said. I think you should do both. I don't think that we're at the point in single cell analysis where we would be able to do the kind of scale of, like, let's say, ChopMed is doing RNA-seq on 15,000 individuals. Single cell RNA-seq is just not there yet. And there are things that we miss there, and that is kind of, yeah, there is no silver bullet in any of these approaches. You should just take multiple.
Right, but even in the bulk sequencing, one can bulk sequence heterozygotes, the millions of loci that are heterozygous and that are in transcripts. And so, is there a value there for looking for cis-EQTLs, or has the bulk sequencing been sufficient?
Yeah, so if you are just referring to an analysis of allele-specific expression from RNA-seq data sets, like that's kind of like half of what my lab does, so I obviously believe in that, and that's the last slide that I showed you. This kind of personalized transcriptome interpretation is all based on allele-specific expression, so yeah.
I will only add to the more general point that it's very important to right now choose the best thing you can do now, but also, and I think Rory said that earlier on, to know when to wait for something that's about to come and to have a little bit of a horizon. This is not, I think, a discussion of what we're doing in the coming year, but more of a discussion of what we're doing in the coming decade, 5 years to a decade. I'm not sure, I'm looking at the organizers. But in the multiple year time scale. And so there has to be some technical flexibility in projects and in efforts like these, and reassessment as you go along, and you start getting, you retire old things and bring in the new.
Yeah. Also, we should remember.
When my lab looked at cis-EQTLs in mouse in public databases via the differential allele expression, many were not real and many were not detected.
Yeah. That may be a problem of the mouse data set, but in GTEx it replicates beautifully. One thing that I want to mention when it comes to single cell analysis and kind of what you lose and what you gain, actually not all genetic variants have very cell type specific effects. There's a lot of sort of like strong, like not just loss of function effects, but other sort of coding effects or promoter effects that are just very strongly shared. And those things you can capture pretty easily from bulk. Easy, cheap, works, scales. And that you don't necessarily need to have cell type specific data to be able to interpret all types of genetic variants.
And just to make one comment about this sort of balance between what to do later and what to do now, I think it's what Eric said in his talk. We spent $3 billion to do the first human genome. We would not have waited for the human genome to become $600 or $300. So at some point we need to be doing something that motivates the questions of what the next technology is going to bring. If we don't do enough of sort of the bulk RNA sequencing, we would not have the motivation to do the single cell sequencing, right? So to some extent, we need to do some of the studies that make a strong case for the technology development and the methodology development, but not too much that it's a waste of money and time, since we can do this again with improved technology. And that balance, I don't think we know exactly what to do, but we should try to find that balance.
Jonathan Pollock, Knight, huh?
I think it's probably
Question. is fixed tissue valuable for RNA-seq, and what are the challenges for actually doing it, and what are the problems of access to it?
I can answer that briefly. I think for bulk RNA-seq, it's valuable. For single-cell RNA-seq, it's work in progress. Technically work in progress. If it works, it would be amazing.
So trying to link these various sessions together and keeping in mind the other Eric's challenge to be specific, should the cohorts / biobanks that are all represented here today, should we be collecting material to make iPS cells for later on all of these individuals? And also keeping in mind we have a fixed budget.
Yes.
Yes.
And collect some blood,
You can
too.
take a vote. Yes.
PBMCs, not whole blood.
That was so nice and quick. I'll give two quick questions. One, Jay, I was really impressed that you could lift over non-coding elements from the mouse into human and show that aggregation. Given the lack of general conservation outside of coding regions, did those elements that you saw the overlap with the GWAS hits have excess sequence homology between human and mouse than average regional non-coding areas?
That is a good question. We did not specifically look at that. And my recollection, if I'm remembering correctly, that even in non-coding regions, the partitioning does tend to go with conserved regions, the partitioning of the heritability. So my expectation is that would be the case if we looked at it.
Cool.
But not because it's a function of synteny. You can lift everything over, but...
No, I know you can lift things over, but I mean, it would be an interesting annotation of GWAS hits in general if there was a pattern there you could tap into for fine mapping. I was just thinking.
I see.
Anyway,
Yeah.
second question for Ramnik. Could you speculate on what type of a sort of community-wide project could take place that could allow massively parallel functional annotation of coding variants?
Yeah, so we could start with two experiments. One is to engineer these variants in primary cells using CRISPR, so that way you can get to certain diseases for where you can't get cells. The other is to take peripheral blood cells from these individuals who harbor these variants and then screen for phenotypes. So one could, right now we, I think, set to sort of look at something like 20 different pathways from things like autophagy to ER stress to various cytokine biology. So I think the challenge is to sort of try to come up with the minimum amount of sample that's needed, but then try to do pathway readouts.
So it'd be great if, as a community activity, we thought about those who would do those assays in their own interesting ways and cell types and so forth could be contributing the screening results back to a central resource.
Can I comment on that, Mark? That I think the challenge today is that the community of people who do a lot of screens and the community of people who know coding variants are not actually intersecting much. And so for labs that actually have great screening expertise and know how to build it, if they want to do something like that with cancer, it's actually really easy. You find the people, you find the data, you know what the variant is, the driver is understood, you know what to do. And I showed an example of that, but it's actually not difficult to do the first step, the informatics step. The lab part is the lab part, but that's separate. I think in human genetics, it's actually really hard to do it well and to do it without making silly mistakes. So it's an opportunity, I think, for this community to try and engage more with that style of functional genomics, which is different than the more observational style. It's actually not the same people in labs.
Yeah,
Okay,
no, you're
well-
absolutely right, and that was sort of my closing thought in
So
the
I was
morning
trying- I was
session
trying to
was exactly
reinforce.
that we need to,
Yeah.
as a community, build those resources so that the durable results from human
Yeah, I'm
genetics
trying to
are
reinforce.
cataloged for the entire community to use.
Okay, and with a few seconds to spare, you now have 10 minutes and exactly, and more than 10 minutes for the break. Let's thank everybody for the speakers.